Natalie, for that great introduction. Hi, I'm Amanda. I'm part of the gene editing team. As Natalie mentioned, we're responsible for creating these gene-edited stem cell lines that are then used for this data collection, the animations and modeling, et cetera. So as Natalie said, we used human-induced pluripotent stem cells for our work. And one of the reasons that we do that is because we just believe that they're satisfying all of the needs for studying normal human cells. So just to remind everyone what these are cells that are derived from a human adult donor as somatic cells that then get reprogrammed into a pluripotent state. These can then, in the lab, be differentiated into many different cell types, which can then be used for a variety of applications, from studying disease, drug discovery, et cetera. Additionally, if you get cells from a healthy adult donor, you can use them to study healthy, normal human cells, which is what we're doing. The benefits are that they are diploid, which means they have a normal number of chromosomes. They're non-transformed, they proliferate well in culture, and they have this differentiation potential, which allows us to do editing in the stem cell state and then differentiate them into the lab, and then we can study the differentiated cells and also the progress that they make as they're differentiating. So to be clear, we don't do any of the reprogramming in our lab. We have an established stem cell line that we use. But we do some of the differentiation. We have sort of a subgroup of our team that specifically works in cardiomyocyte differentiation currently, which is the first cell type that we're interested in studying. We make all of our edits using CRISPR-Cas9. This is a molecular scissors that allows us to very specifically cut the human genome. And in our case, we're inserting a large piece of DNA that encodes a fluorescent protein. We put this either right at the beginning or right at the end of another protein sequence. And so that way, when that protein is made, the fluorescent protein comes along for the ride, essentially. And we get these endogenously regulated fusion proteins, which are absolutely perfect for live cell imaging. They have many benefits over some of the traditional overexpression methods. So as Natalie mentioned, we do just a lot of quality control on the lines that we generate to make sure that they're going to be the highest quality possible so that we're getting the highest possible quality data out of them. So just to kind of give you a high-level overview of that, we'll go to our website, which is allencell.org. If we go to Cells and Cell Catalog, the first thing you'll see is a list of all of the cell lines that are currently publicly available. We'll have 20 currently available for distribution by the end of the year. If we look at one of them, which is nuclear lamin B1, this is tagging the nuclear envelope of the cell. So again, here's a Z-stack going from the bottom to the top of the cell, showing that beautiful nuclear membrane. And this is all live imaging, artifact-free because it's all endogenously expressed. We just get these really crystal clear, beautiful images. Also, because it's live imaging, you can look at a time-lapse video, which shows these really beautiful colony dynamics. You can see cells dividing in real time. Of course, this is not real time, but you can see them dividing. At the bottom of the page, we're showing the editing design, including the CRISPR we used, and then all of the quality control. So did the GFP get in the right place in the genome? Is the protein behaving properly? Is it localizing properly still? And of course, we're using stem cells. We want to make sure that they're maintaining a pluripotency state. So we do a variety of pluripotency analyses on the cells before we release them, both for imaging and for distribution. Our protocols can be found if you're interested in growing one of our cell lines, or if you're interested in making a similar edit in a different cell type, maybe another one that you're studying. You can obtain the cell lines through our distributor, which is Coriell. They distribute a number of stem cell lines. And then additionally, like I said, if you're interested in making your own edited cell line, you can get the donor plasmid that we've used to insert the fluorescent protein through AdGene. So of course, making these tools available to the community is part of our mission, but as Natalie mentioned, we're really interested in imaging these cells and getting data out of them. That's what Irina is going to talk to you a little bit later, some of the data and interesting information we're gathering, and then Dave will show you how we're making models from that as well. So Natalie mentioned it, and I mentioned it earlier, that we're making cardiomyocytes out of our stem cell lines now. So here's, this is the stem cell line. There's a couple dividing cells. This is alpha tubulin, which the cytoskeleton is labeled here. And if we differentiate this into a cardiomyocyte, we can see this beautiful reorganization of the cytoskeleton. And of course, this is all live imaging, and the cardiomyocytes, of course, beat, so we can see this beautiful dynamic video that we've got from this experiment. So this is an example where the protein of interest that we've tagged is expressed both in the stem cell and in the cardiomyocyte. But But as we're interested in studying different cell states and cell types, we are interested in also looking at proteins that are specific to cardiomyocytes. This is one example of one. This is troponin. This protein localizes to the sarcomere, which is sort of this railroad track-like structure that's going to actually expand and contract as the cell beats. So just another really beautiful example of very clear imaging that we're getting from these endogenously tagged lines and some of the benefits from using the stem cells and doing the live imaging. Another way that our stem cells have been used in the community, this is data from the Friedman lab at UW. They work on kidney organoids. So these are all 3 different kidney organoids that they've made from 3 different of our cell lines, and this is just at varying magnification. So this is the nuclear envelope. This is DSP, which is cell-to-cell junctions. And this is, of course, the cytoskeleton, and you can see that they've also been able to capture a really beautiful dividing cell in their tissue. So being able to study these proteins at the tissue level is something that they haven't been able to do before, and our cell lines have really enabled them to get a better understanding of how the tissues are functioning, and hopefully will also help them understand disease processes better. One thing that whenever we're telling people that we're editing stem cell lines, they get excited about, well, can you put more than one edit in the stem cell line? Because wouldn't it be cool to see 2 in one place at the same time? And we agree, it would be great. So this is something we've been working on, as Natalie mentioned earlier. We have an example here where tubulin is labeled with a red fluorescent protein. And then we have the endoplasmic reticulum in green. And so when we merge the 2, we can now see 2 endogenously expressed proteins in the same place at the same time, in the same live cells, and see how they both change during, for example, mitosis. So I'm going to hand it over to Irina now. She's going to tell you a little bit more about how we're gathering data and learning about these cells. And then after she's done, Dave is going to tell you a little bit more how we're taking data from our individual cell lines and integrating it into a single model, while the gene editing team works a little more on creating more of these dual lines.
My name is Irina, and I have the honor to represent the assay development team at the Allen Institute for Cell Science today. And I'm really excited to share a quick glimpse into the mystery cell population with you. So Natalie already showed you that image of the team organization of the Institute. And once the cells are made and validated by the gene editing team, the microscopy team images those cells on a standardized automated imaging pipeline in thousands of replicates, together with a plasma membrane dye and a DNA dye, so we can use the plasma membrane and the DNA as reference structures. Once those cells are imaged, they're being segmented, and all the good segmentations are uploaded into the Allen Cell Explorer. I don't have a lot of time today to actually really show you the 3D cell viewer, but I highly encourage you to actually go and explore the cell. It's really fun. You can spin the cells around and slice through them. And currently, we have 20,000 segmented cells in our viewer. You can both see the structure and the DNA and the plasma membrane. And one of the things that we're doing at the assay development team is actually exploring the data, and so can you. So if you want to go to our website, allencell.org, go under Tools and Data, you can go to this 3D cell viewer. Today we're just going to look at single cells and just at our mitochondrial line, and I'm just going to click really quickly through the image gallery. And even without going in details, I hope you can appreciate the huge variety of membrane or cell morphologies, where the nucleus sits and where the structures are in the cells. And so one of the questions that we had is, where is this variation coming from? Why do we see these large amount of shapes, even though in theory it's the same gene-edited cell line? And a partial answer to these questions are we getting when we're using our plotting tool, and you can plot a variety of variables against each other. Today we're just going to plot the cell volume versus the nuclear volume. And when we look at all our structures, as Natalie already as Natalie already told you, we can see the two populations. The upper population is all non-dividing cells. And if we look at the lower populations, we notice that these are all dividing cells. So as our first project, we wanted to understand the physical organization of the dividing stem cell. To do so, we had to characterize our cells into dividing cell cycle stages. And so we took all the DNA images that we got from the pipeline and said, okay, can we sort our cells into cell cycle stages based on DNA characteristics alone? And from there, we identified 8 stages, non-dividing stage and 7 dividing stages. And when we sorted our microtubule line into those 8 cell cycle stages, we saw that they beautifully match the biological relevant mitotic stages. So we have one non-dividing stage, or interphase. We have 2 stages of prophase, 2 stages of prometaphase, 1 stage of metaphase, 1 anaphase, and a combined stage of telophase and cytokinesis. Now that we have the stages, we manually annotated about 9,100 cells into these stages, and we identified 700 mitotic cells. And we're really excited about these data sets because we have endogenously tagged gene-edited lines. So we have a stable cell, it is non-transformed, and we have an endogenously tagged protein that we now can look at. So that means we don't have any overexpression artifacts. Also, we have 14 major organelles and key intracellular structures in our data set. So instead of looking just at 1 or 2 structures at a time, we can look at a large variety of organelles and structures in the cell and hopefully integrate them to really understand the physical organization. Now that we have the data set, we can ask 2 questions. What happens to cell morphology and localizations as the cells divide? And what happens to the structure as the cells divide? Let's look into the cell morphology first. When we look at transmitted light images of a colony over time, we can see that the dividing cells round up, let me go back, round up, okay, they're not rounding up anymore, but you saw that they were rounding up and rising as the cells were dividing and then kind of settling back into the colony when they're done dividing. So we wanted to analyze the cell roundness, quantify the cell roundness, and we did this by dividing the shortest axis of our cells, which is this axis, by the longest axis of the cells, which is this axis. And the closer the number is to 1, the rounder the cells. And when we look at the cell roundness over our mitotic stages, we can see that starting at stage 3, or the early prometaphase, we see a trend of the cells to round up, and then they continue to round up through the later stages of mitosis until the last stage of mitosis. Since the cells were rising up, we also wanted to see and quantify their position within the colony, which we did by looking at the cell center mass relative to the bottom of the cell colony. And there we see the same trend that at cell cycle stage 3, or early prometaphase, the cells starting to rise up and then kind of stay elevated as they continue dividing. So the question is, how can those cells undergo such a dramatic morphology change, but still be in part of the colony? And an answer to this might be from this movie, where you have a plasma membrane gene-edited cell, and all we see is the top slice of the cell in green and the bottom slice of the cell in magenta. And I hope you can appreciate how rigid that top part of the cell stays as the cells kind of explores, while the bottom part of the cell like vastly explores the surroundings. We see the same when we look at the tight junctions. So this is a maximum intensity projection, and we kind of look from those tight junctions into the cells. You can see that in interface, when the cells are not dividing, the membrane is closely associated underneath the tight junctions. But as the cells divide, the membrane rounds up underneath the tight junction, and this is really noticeable at our last stage of mitosis, where this is the tight junctions between this dividing cell with its neighboring cells, and underneath you can see this huge bulging of the plasma membrane. So we think by maintaining the contacts with the neighboring cells, this is how the cells, once they divide, can maintain their position within the colony while rounding up. So they're maintaining here their contacts with their neighbors, anchoring in this colony, while underneath they're significantly rounding up and rising up within the colony. We can also look at what the structures are doing as the cells divide. And this is kind of an overview of all our 14 key organelles and structures. And we can do the classical approach, and we can look at one single structure over time to understand the dynamics of that structure as the cell divides, or, which I think is more interesting and it's going to be really exciting to do so, we can pick one distinct time in the cell cycle and look at all the structures together and integrate them to really understand the organization of that cell at that given time point. I don't really have time today to talk about all the structures, but I really want to point out that in the early prometaphase, or cell cycle stage 3, where we saw the cells round up and rise up, this is also where we see the most dramatic changes in the structural organization. Today I'm just going to focus on 3 structures, and their theme is going to be structures that partially or totally disappear during cell cycle. The first one is well studied. It's lamin B1, or the nuclear envelope. And we can see the beautiful envelope, and then again at our mitotic stage 3, or early prometaphase, we see a partial disassembly of the nuclear envelope, which stays disassembled until telophase cytokinesis, where we can start to see it reassemble around the DNA. And based on time-lapse movies that we took of the lamin B1 gene-edited cell lines, we can even differentiate that interface into 3 more interface stages. We have an early one where we see lamin B1 spots within the nucleus and outside the nucleus. We have a mid-interface stage where we just see a nice nuclear envelope, and we have a late interface stage where we start to see laminar invaginations. Another structure that completely disappears is the Golgi. The Golgi kind of forms around the nucleus, so we have the nucleus in the cell, and we have kind of one laminar structure that comes, or one tubule that comes up, and then we have a beautiful laminar structure on top of the nucleus. And that laminar structure starts to fragment in the early phases of mitosis, and then again at our mitotic stage 3, or the early prometaphase, the Golgi totally disassembles, and we do not see any Golgi until the last stage of mitosis, when the Golgi reforms. Another interesting structure is the nucleolus, where we go from a foci nucleolus structure of fibrillarian to a phase transitions, where fibrillarian is like in this melted structure in our mitotic stage 3. Then fibrillarian completely disappears and then starts to reform at the 2 later stages of mitosis. But interestingly, it's actually reforming on the sister chromosomes and not in the nucleolus. So I talked a lot about actually what the structures are doing in this timeline, and Dave is going to show you how we wanted to actually integrate all the structures in this line.
Hello. Oh, this does work. Hi, so I'm Dave Williams, and I'm part of the modeling team at the Allen Institute for Cell Science. And the goal of the modeling team is to try and help us see more with the data that we've gathered. So we can already see a lot. We can't quite see this much. This is a totally fake composite image. No image actually looks this great. But we can see a lot. We're limited, however, in a couple of ways. And one of those limitations is bandwidth. There's only so much bandwidth of light that you can shine into a cell. And once you've excited fluorophores, there's only so much light that you can receive back out of the cell. Additionally, there's a photon budget. If you shine too much light in, the cell dies and you can't see anything else from it. So wouldn't it be nice if there was a way we could use all the images that we've gathered to see more, perhaps to see more structures at one time? So there's two ways that we can go about this, or two ways that we're currently going about this. One is to try and see more structures in a single field of view, try and see more things in a single measurement. And the other is to combine multiple measurements to see more structures at the same time. Let me talk about the first one. So there's information in schmutz. I think of schmutz as like the stuff that gets like stuck on my glasses, but for the purposes of this, we can say it's any structure in an image that you don't really know what it is. You can have an image where there's obviously a lot of structure to it, there's obviously something going on, but it's not really interpretable to me until I find the right transform. And then I can take the information in that image and create a version of it that's really obvious to me what's going on. So what information is hiding in our transmitted light images? What information is there that we can't see? Well, there's a couple ways of getting this out. The most traditional way is to actually physically perturb the image, is to introduce a dye or a fluorophore. And that obviously can highlight a structure. This is a nuclear dye, and it's letting us see the DNA in the nucleus quite well. But that's not the only way of seeing that, because if I look at this and I eyeball it, and I sit down at a laptop and I spend 20 minutes being really bored trying to highlight the nuclei, I can do an okay job of it. So is there a way that we can encode that intuition that I've developed about that image into a model and have it do that highlighting for us, but faster, better, cheaper? Well, yeah, you can train a model to detect information in schmutz. You start out with many small segments of your transmitted light image, and then a series of ground truth images, which are the dye images for those same fields of view. And you feed those into a UNetish model, which is a neural network model that is described more on the website and also in the paper that accompanies this work. And initially it puts out something kind of blurry, but it's a model, so you give it an error function, and it slowly learns from the difference of the output and the ground truth how to produce a better image. How good of an image does it produce? Well, it works pretty well for nuclear predictions. If this is our bright field image, and this is our dye image, our ground truth, this is our predicted image. It's reasonably good. In fact, it even does a little bit of denoising because that noise isn't really present in the bright field image. This works very well for structures that are dense or have membranes, things that produce a lot of information, a lot of schmutz in the bright field image. So for nucleolus, it works great. Again, the middle image is our fluorophore image, and the right image is our predicted image. It doesn't work so well for diffuse or filamentous structures. They don't necessarily leave as much of a trace in the bright field image, and it's harder to predict them. So with this, we can see things that we never could before. We can look at a long time lapse and see 5 or 6 different structures in that time lapse image, all highlighted. That's pretty cool. However, it's still limited. There's still things this can't do. Every model is good at some things. This is good at some things, but not everything. What about structures that don't necessarily leave a lot of information in the bright field image? Well, that's where we come to another approach, which is to use as an input, instead of bright field images, images that have cell membrane and nuclear dye. And really what we're asking here is, can we find the things that we have for some cells, where we have, each cell has like lamin B or fibrillary, and it has one of these things. Given what we have for every cell, which is a dye for the cell membrane and a dye for the DNA. The formalism of this is just, can you tell me the probability that I'm going to have a given structure given an input cell and nuclear image? And yeah, again, we can train a model for this. The input is our cell and nuclear images. We encode that, we compress it down into a reduced dimensional space. This is the same thing as running it through a very, very, very specific zip program. And that represents all the structure that we expect to see in the cell in a very small dimensional space. And then again, you can expand it, you can decode it back into a representation of the cell and nuclear images. That gives us our first error function for this model, the first thing that we're training it to minimize, which is the input images minus the output images. Now, additionally, we want to be able to explore this reduced dimensional space a little bit more easily. We'd like it to be smooth. We'd like it to be continuous. So we take a normal function and add a normality error. If the reduced dimensional representation isn't very smooth, the model gets penalized. So it learns how to make a nice, smooth representation. Now, the third error function for this model is just a way to goose it a little bit, which is to train an adversary that gets to see both the input images and the output images and try and tell which is which. If it's good at telling an input image from an output image, that means there's an obvious difference between them, at least an obvious difference to the adversary, and that's a penalty for the model. It should be hard to tell the difference between those two images. So we wrap this section up into a single model. We say that's our cell and nuclear shape model. Now, we can repeat the same training process using this for the cell and nuclear shape, but also including the structures, and do this again one structure at a time to learn a reduced dimensional representation of the structures as well. If we combine these, now we have a model of structure, cell, and nuclear images. Again, this is represented as saying what's the probability of this structure given a cell and nuclear image. And we can say, okay, let's not look at the probability of any structure. Let's just look at the one that's most likely given a given input image. And do that for a specific structure. Let's train it initially for fibrillary, later for alpha-tubulin, lamin B1, TOM20, and produce a set of models that can predict any given structure given an input cell and nuclear image. Now, the output of this is this grid here, where this column here are observed cell and nuclear images and observed structural images. This top row are input cell and nuclear images, and everything else is a predicted structural image from that cell and nuclear image. And it's pretty hard to actually tell the predicted structures apart from the observed ones, and that's a reasonable measurement of how well the model's doing. So that's it for the cell science talks. We'd like to thank the team at the Cell Science Institute, the rest of the Allen Institute, and of course our founders. And at this point, I think, take any questions.
Yes. There's a question over there.
I just wanted to ask you whether you think the rules are going to be general across cell types.
That's a great question. So, no, this has to be retrained, not only from cell type to cell type, but from imaging modality to imaging modality. If you substantially change your microscope settings, that'll change how those images come out, and you'll have to retrain your models. Pick up the mic.
Question there.
Yeah, I had a question for the last speaker about the part where you show that the during mitosis cells are becoming rounder in kind of an anisotropic way. Does that have to do with their orientation relative to gravity or relative to the kind of surface that they're on? So when you say top versus bottom, what do you mean by that?
Well, yeah, top versus bottom is the bottom is the substrates the cells are growing on, and then those cells are growing in a one-layer epithelial sheet, and they have like a clear epithelial orientation. So you have stress fibers at the bottom, and then on the top of the cells you see an actin ring and all the cell contacts and cell junctions. So in this way we have like a polarity with the top and the bottom.
I see, but if they could kind of expand in either direction, do you think that there would be a polarity to it?
They do in your body anyway.
Yeah.
Yeah, they do. I don't know. We could check that if gravity has anything to do with that, but my wild guess would be no.
I think the standard assumption is gravity doesn't matter at the scale of a cell.
Okay. Yeah, this is a neurobiologist asking a cell biology question.
Another question.
What's the relationship between the amount of the different proteins and their subcellular distribution? When you see structures disappear, is that because the protein that constitutes that structure is gone, or is that because it's diffused around the cell so that it doesn't show up in the images?
So for Golgi and for the fibrillarin, if we would actually dramatically increase the contrast, you kind of can see the GFP diffused in the cell. So I think the protein is still there. It's just not in its original structure.
They reorganize so quickly.
Grab the mic.
without their
Okay, no further questions. Let's thank again our team from Cell Science.